Hello students, today's video is about the transportation of material across the plasma membrane. This is the second part. We have already studied about the passive transport system in our previous video. Before watching this video, make sure that you must be familiar with the structure of plasma membrane and fluid mosaic model and the transportation of material across plasma membrane, that is passive transport. I have made both a video. You can go to the library and find them for your better understanding. So today's topic is, we have to study two important topics. First is active transport and second is vesicular transport. So let us start with the active transport. What is active transport? Active, as the name suggests, it is against the concentration gradient and so that it requires energy. In active transport, the molecule transfer from lower concentration to higher concentration because it is against the concentration gradient, so it requires energy and it mediated by the carrier protein, and required energy is in the form of ATP, or in case of plant cell, in case of photosynthesis, it is light energy from the sun. It results in the accumulation of solute on one side of the membrane because it is against the concentration gradient. It pushes the molecules from lower concentration to higher concentration so that the concentration of molecule is accumulated in one side of the membrane. If we look at the type of active transport, it is of two types. First is primary active transport and second is secondary active transport. So what is primary active transport? The primary active transport is also known as active transport because it directly uses energy to transport molecule across the membrane. These charged particles require ion pumps or ion channels to cross membrane and distribute through the body. Substances that are transported across the cell membrane by primary active transport include metal ions such as sodium ion, potassium ion, magnesium ion, and calcium ion, etc. The example of the primary transport is sodium potassium bond, which is a primary ATPase universal to all animal cells or all animal life, which help to maintain the cell potential. The sodium potassium pump maintains the membrane potential by moving 3 sodium ions out of the cell for every 2 ions moved into the cell. If you look at the structure of sodium potassium pump, you will find that sodium potassium pump in this configuration has higher affinity for sodium ion. Sodium ion from the cytosol attached to the transporter. Then ATP is attached to this transporter and bring the conformational changes, and the affinity of the sodium is decreased, so it flips its sodium ion out of the cell. This ATP is converted dephosphorylated or converted to ADP, and when the phosphate is removed, ADP is formed. The conformational change bring the transporter in such a position that its affinity for the potassium ion is increased, and this transporter is flipped in this way so that it releases 2 potassium ion inside the cytosol. Look at here, The concentration of sodium inside the cytosol is already low, but these are pushed across the membrane to outside, and it accumulates the sodium ion in extracellular space, while as instead of 3 positive ions, only 2 potassium get the entry into the cytoplasm. So here develop the positive charge, and inside the cell, the inner side of the cell develop the negative charge. In this way, cell maintain the potential of the cell membrane. If we look at the other examples of ATPase transporters, the most striking example is P-type ATPase. One of the pump is sodium potassium pump, which we have studied now. Other example is calcium pump and proton pump. Then we have F-type ATPase present in mitochondrial ATP synthesis is the example which take part in the ATP synthesis, and F-type ATPase are also present in the chloroplast, where they are also take part in the ATP synthesis by photophosphorylation process. B-type ATPase are present in the vacuolar ATPase. They are present in the vesicle of vacuole of the plant cells, where they maintain the fluidity, maintain the pH and the turgor pressure of the cell. Another example is ABC transporter, that is ATP binding cassets. The most striking example of ABC transporter are MDR and CFTR. Now the second is secondary active transport. Why secondary? Because in this transport, energy is not directly used. Instead, secondary active transport is a method of transport in which the electrochemical difference is created by pumping ion out of a cell is used to transport molecule across a membrane. Here, in this process, still consume ATP to generate that gradient. The energy is not directly used to move the molecule across the membrane, hence it is known as secondary active transport. What happened in this? If you look at what happened in first step, primary transport system transporter, by using energy, push the molecule across the membrane, which cause the higher concentration of this molecule in the extracellular fluid. Because the higher concentration of these molecules in outer side of the extracellular fluid, so there develop a concentration gradient, because there is a difference between the concentration across the two sides. So what happened in next step? These molecules re-enter through the channels across the concentration gradient. In second step, it doesn't require energy, but to generate this concentration gradient, primary transporter use energy for the generation of this gradient. So secondary active transport uses energy in indirect way, so it is known as secondary active transport. Carrier protein for active transport. There are three types of these protein or transporter. First is uniporter, second is symporter, and antiporter. Uniporter are the carrier proteins which transport a single molecule across the membrane. The direction of the movement maybe from outer side to or from inner side to outer side, but it only transports one molecule at a time. Other type is a sym-porter carrier, where two different ion or molecule both in same direction are transported, like this way or like this way. The most striking example of this is sodium glucose pump, where glucose and sodium are transported. And third is antiporter. What happened? One molecule move across the membrane to one direction and other molecule move in different direction. For example, if A molecule move inside the cell, then B has to move outside of the cell. This is the type of antiporter cell, antiporter transport. Now we have vesicular transport system. The first is endocytosis and second is exocytosis. Endocytosis is a type of active transport that move particles such as large molecule, parts of cell, and even whole cell into the cell. Endocytosis consists of three types of endocytosis, that is phagocytosis, pinacocytosis, and receptor-mediated endocytosis. Endocytosis take particles into the cell that are too large to passively cross the cell membrane. Because we have channels and ports by which we can transporters which can transport a smaller molecule from one side to another side, but the bigger molecule cannot cross through these transporter and channels. So what happens? Their endocytosis play an important role. The plasma membrane of the cell invaginates, forming a pocket around the target cell. The pocket pinches off, resulting in the particle being contained in a newly created intracellular vesicle formed from the plasma membrane. So there are three types of endocytosis. First is phagocytosis, other is pinacocytosis, third is receptor mediated endocytosis. What happens in endocytosis? This large molecule, when it has to be enter into the cell, it attaches to the plasma membrane. Plasma membrane forms vesicle around this molecule. Eventually, this phagosome get pinched off from the membrane and move inside the cell. This membrane regenerated, because we know that plasma membrane is a fluid in nature, and inside the cell we will study this, what happened to these phagosomes. We will study when we study about the suicidal valve of cell, we known as lysosome. When we study about lysosome, we will study what happened to these vesicles inside the cell. For now, let us know about the phagocytosis. Phagocytosis means the cell eating. It is a process by which a large particle such as cells or relatively large particles are taken up by the cell. What is the difference between phagocytosis and pinacocytosis? Pinacocytosis is a cell drinking. Here pinacocytosis take the liquid particle. What happens? Phagocytosis take the solid particle, while as in pinacocytosis the fluid molecule get into the vesicle, and this vesicle pinch off from the cell membrane and get entry into the cell. The difference is that in phagocytosis the material is in solid form, and in case of pinacocytosis it is in the form of liquid. And third type of endocytosis is a receptor mediated endocytosis. Here the molecule has a receptor on the plasma membrane. Specific molecule has specific receptor. what happen? These molecules attach to these receptors, and when these molecules attached to the receptor, is invagination of plasma membrane take place, and there are the protein known as cleftin proteins which coated the vesicle, and this vesicle pinch off from the membrane and transport these molecules inside the cell. So look at it here. It is a solid particle which attached to the membrane. Membrane get invaginate, then eventually this pinched off and form a phagosome. This is a phagocytosis, while as in case of pinacocytosis, these are the leucal molecules which get entrapped into the membrane, and membrane invaginate, finally form a phagosome and transport the leucal material into the cell. Third type of endocytosis is receptor mediated, in which this is the material which has a receptor here. It attached to this receptor, and from receptor a cleftin coated protein coated this vesicle, and vesicle move inside the cell. This is known as receptor mediated endocytosis, where the molecule is attached to the receptor. So receptor mediate the endocytosis. So with next is exocytosis. Exocytosis is the process where the waste material of the cell is transported, waste material of the cell synthesis is transported outside the cell. In exocytosis, the cell create a vesicle to enclose something inside the cell for the purpose of moving it outside of the cell across the membrane. This most commonly occur when a cell wants to export an important product such as cell that synthesize and export enzymes and hormones that are needed throughout the body. For example, take an example of beta cell of the pancreas. Beta cell of pancreas synthesize the insulin, but insulin is not used by the beta cell of the pancreas. It has to be exported outside the cell so that it can be used by the body's other cell. So what happens? This insulin molecule are get entrapped into vesicles. These vesicles are transported to the plasma membrane where they get fused, and what happens in next step, the plasma membrane these get open, get invaginate in such a way that it transfers these molecules outside the cell. So endocytosis and exocytosis play a very important role in the movement of the material across the plasma membrane. So with this we end our video. Sorry, before that let us study about the difference between active transport and passive transport. As I already told you that active transport moves substances from region of lower concentration to higher concentration, that is against the concentration gradient, while as in case of passive transport occurs naturally as substance moves down a concentration gradient. Active transport requires energy as it does not occur naturally in the absence of active force, while as energy is not required in case of passive transport because the movement is with the concentration gradient. So with this we conclude our lecture. We have studied about the transport across the plasma membrane in detail. So we shall meet in next video with new topic. Till then, take care. Thank you.